Good afternoon. My name is Eric Soler, CBO of Outpace Bio. Outpace is a clinical stage, privately held biotech. We are reprogramming cells for solid tumors. We're focused on a critical gap in this space. There's been a lot of recent efforts to engineer cells outside the body to optimize manufacturing. At Outpace, we use AI-driven protein design to design cells to have optimal function inside patients. We think that's a critical step to making cell therapies that are safer, more effective, and can be delivered at scale. Our co-founders have been at the forefront of protein design and engineered cell therapy for the better part of a decade. Mark and Scott met in David Baker's Institute for Protein Design. They joined the field at a time when proteins could be designed to fold into the desired shape, but introduce dynamic parts, precise interaction specificities, and ultimately created novel biological function. They got to know Stan Riddell at the Fred Hutch along the way and began collaborating and realized that protein design and cell therapy had a lot more to offer each other. That insight led to the founding of Lyell Immunopharma and ultimately spinning out Outpace to scale our technologies into the cohesive pipeline that we're advancing today. I think everybody in this room probably appreciates the tremendous impact that CAR T-cell therapies have had on the treatment of blood cancers. Here's the CAR data where, more than half of patients are still in remission, a durable cure. And what's interesting about that is that it coincides with CAR T-cells that stick around for months or even years. When you look at the bottom left in solid tumors, this is a completely different story. 90% of cancers are solid tumors, and experimental T-cell therapies like Gavo-cel, which is shown here, do lead to significant tumor regression in patients. But unfortunately, when you look at this chart, you realize that those T-cells are gone in about a month. And of course, when you increase dose or dose escalate, you often run into dose-limiting toxicities. Now, it's not surprising that a one-and-done therapy that doesn't stick around can't prevent patients from progressing. And we know very, very well what the problems are here. T-cells don't get into solid tumors very well. If they do get in, they don't survive well. They rapidly inactivate. They're susceptible to antigen escape. And again, high doses drive toxicity. So what we're doing at Outpace is really aiming to take this PK curve and extend it and make it look more like what we know leads to really durable activity in blood cancers. Our approach is structured around these four pillars. The first thing that we do is we design the optimal components to attack the cancer with maximal potency, specificity, and safety. This includes best-in-class CARs, TCRs, logic gates, and safety switches. We also use our Outlast regulators to preserve the ability of our T-cells to persist and to avoid T-cell exhaustion and preserve stemness. We have a suite of Outsmart designed cytokines that are secreted by our cells to broaden the immune response in the local tumor microenvironment and prevent antigen escape and other immunosuppressive pathways. And then finally, we have a novel in vivo delivery approach that we call Outdrive that permits cell type specific targeting. We're able to achieve high transduction with minimal immune activation, and we look at that as a safe and effective way to scale the delivery of these technologies. Now, what's particularly powerful about the approach that we take is we can combine multiple proteins together in a single lentiviral vector, and that allows us to make, at the end of the day, a very simple and manufacture a very simple product. How are we able to take that ambitious approach? Well, as I mentioned before, it's driven by AI-powered protein design. Mark, Scott, and David have been advancing this field for many years. We start by coming up with a function that we'd like to see in our cells. We use an initial generative AI model called RF diffusion to design the ideal backbone for that. I think, as everybody in this room probably appreciates, the structure of a protein dictates its function. We then use a second generative AI model to assign the amino acids to fill in that backbone structure, and then a third separate generative AI model to validate in silico that what we develop actually matches our initial design. What comes out of that design process is a library of modular protein parts that I'm showing here that we have built, characterized, and integrated into our therapeutic pipeline. On the left, you can see the four technologies that are on board our lead program, OPB-101. I'll walk those with you in more detail in a moment. In the middle, you can see two completely different technologies on board our second program. This is important for us to minimize the correlated technical risk between our pipeline programs, to test multiple hypotheses in the clinic, and ideally, to be able to iterate based on clinical insights to develop better technologies in the future. On the far right, plenty of technology assets that we have ready and characterized for additional pipeline programs and for our partnering efforts. Now, perhaps the best way to explain how all this works is to walk through our lead program, OPB-101. It's a mesothelin-directed CAR T-cell therapy. It incorporates four of our foundational technologies. The first at the top left is our outspacer optimized mesothelin CAR. Our outspacer technology allows us to form the perfect immunological synapse between the CAR T cell and the tumor cell. This is sensitive down to the level of a few angstroms, and prior attempts in the field were way off. In doing so, we're also able to design a binder that is not susceptible to shed monomeric mesothelin. That has been a big issue for ADCs and T-cell engagers that have tried to go after this target previously in this space. The second thing that we bring on board is our Outlast OP1 regulated promoter. This autonomously programs the cells to rest and avoid T-cell exhaustion so that they can continue to maintain their activity. We use that regulated promoter to also control the secretion of our own designed IL-2 cytokine, secreted only by the cells in the tumor local tumor microenvironment. Outsmart designed IL-2 is designed by us to signal with several orders of magnitude more activity on the T effectors, TILs, and NK cells in the tumor microenvironment while avoiding interacting with T regulatory cells and lung epithelial cells. And lastly, we have on board our OutSafe EGFR op safety switch, which is, leads to rapid ablation of the T cells in response to cetuximab. Now, importantly, on the right-hand side, you can see some of our in vivo data. We use very stringent lung xenograft models here. These are H1650 lung adenocarcinoma. You know, just orienting you here, in red we have our, in red we have our best-in-class CAR alone at the top. You can see at 2 million cells, we're not able to control the relatively large tumors that we're dosing here. We then drop the cell dose by an order of magnitude, so 200,000 OPB101 T cells. What's important is the shape of the curve. Over a number of days, those cells do not control the tumor. It continues to grow. Eventually, the tumor is melted away by the OPB101 T cells. We've cured every single mouse we've ever dosed with 200 T cells. We've pushed that even further in the bottom left. You can see 50,000 T cells are eliminating tumors that grow up to about 1 gram of tumor tissue. That's roughly 1 billion tumor cells being eliminated by an initial dose of 50,000 T cells, so a 1 to 20,000 E to T ratio. So impressive kinetics, but we're particularly interested in the mechanistic data behind it. When you look at peak tumor volume, you can see that the OPB101 T cells have about a 300x increase in intratumoral CAR T cell count. So we're solving trafficking, we're solving expansion. And when we phenotype those cells, we see a dramatic reduction in PD-1, TIGIT, checkpoint markers, and T cell exhaustion markers. So the kind of mechanisms of dysfunction from earlier that we're looking to solve. We've looked into the technology contribution of each of these components. The too long didn't read version of this is that each technology component that we bring in adds about a 10-fold improvement in the potency and also about a 10-fold increase in the expansion capability that these cells have in the peripheral blood. Our lead indication for OPB101 is high-grade serous ovarian cancer. This is a relevant xenograft model, intraperitoneal SCOV3 injection. We put the OPB101 cells in through a TIL vein injection. And you can see again from 50,000 cells up to 500,000, very nice reproducible tumor killing, and that same OPB101 expansion that attenuates once the tumor is cleared. We noticed in our earlier studies that OPB101 cells maintained through their tumor killing interesting stem-like markers, CD45RA, CD62L. These are hallmarks of long-term proliferative capacity for T cells. And we wanted to really put these cells to the test and see if in a very stringent and robust serial passage model, could they maintain that? Could they maintain their activity? What you see here is the same H1650 model that we used before. The left-hand column should look very familiar in terms of the killing kinetics. The cells get into the tumor at a relatively low dose. They expand, they persist, and they melt that tumor away. We then isolated surviving OPB101 T cells from that cured mouse. We put those cells directly into another established tumor, and you can see in the middle column there that, again, we see really nice expansion, persistence, and tumor killing with attenuation of that expansion over time after the tumor is cleared. Now, importantly on the right, the phenotypic data shows us that about 1/3 to 1/2 of these cells are expressing that really important stem-like phenotype. And to put that simply, we think a lot of people are engineering their cells to try to create a dam to preserve the stem-like phenotype. And when those cells hit the tumor in vivo, the dam crashes and all the cells terminally differentiate. It appears to us that our transgene is able to act more like a water gauge, where a steady stream of effector cells come out for really potent tumor killing, which we've shown you here is durable through really robust serial challenge, while preserving and maintaining that really important stem-like phenotype. Our second program is OPB201. It's a PRAME-directed TCR T therapy. It has 2 completely orthogonal technologies on board. The first is the outmatch optimized PRAME TCR. This is a technology that allows perfect pairing of the alpha and beta chains of the TCR. It does not require endogenous TCR knockout. The implication of that is dramatically improved potency compared to PRAME TCR benchmarks and dramatically reduced complexity of manufacturing. The second technologies are Outlast TF1 regulator. This is a design-targeted degrader technology that binds to all three members of a pro-exhaustion transcription factor family, and it functions a lot like PROTAC without the need for small molecule control. What we're showing here are the same stringent H1650 xenograft models, and again, you can see from multiple donors shown this time that the same kinetics of that curve, where low doses of OPB201 cells in this case are able to infiltrate the tumor, expand, persist over time, lead to very reproducible killing, expansion of those cells, they contract after the tumors are cleared, and then there's that maintenance of a really nice memory T pool after after tumor clearance. We've just talked through what we're doing in this horizon of our company's growth, which is aiming to demonstrate durable efficacy in solid tumors with OPB101 and OPB201. That'll be driven by our Outlast, or Outsmart, or Outspace, or Outmatch technologies. Importantly, we're using single lentiviral vectors, and we're using established autologous manufacturing approaches. Now, that's the foundation that we're hopefully going to be building on. In the future, we turn our eyes towards scalable access, and to do that, we have our own AI-designed in situ delivery program called OutDrive. We're really hoping to use that to solve some of the key issues that we see in the space right now, and to bring along these validated technology assets so we can now drive really exciting engraftment, expansion, and persistence of T cells. Now, there's been a tremendous amount of activity in the in vivo CAR T space recently. I'm sure all of you are aware of that as well. We think people are perhaps overlooking two limitations with respect to existing solutions. The first is the perceived trade-off between COGS, or high transduction efficiency, and safety, or the potential for systemic cytokine activation. And the second is that we don't believe that CARs alone will be sufficient or adequate to drive the kind of efficacy that we want to see in lymphoreplete patients. At Outpace, we believe that you need to be better than autologous, not just as good as. And similar to how we usually approach things, we're really using AI-driven protein design to address all these issues at once. We're working off of a lentiviral backbone. It's what we understand extremely well mechanistically. It has a tremendous amount of safety data behind it, and it's effective. And really what we're doing is using OutDrive to go after safety by ablating native tropism, redesigning that LVV synapse, and in doing so, allowing ourselves to specifically target T cells in vivo and reduce systemic cytokine release. We go after COGS by boosting transduction and co-stim signals, including molecules that will also mitigate host restriction factors, and in doing so can boost transduction efficiency. And then, as I mentioned before, bringing along our, hopefully validated in the clinic by the time we're pushing this forward, technology assets that will drive the engraftment, expansion, and persistence of these cells in a lymphoreplete environment. We are currently advancing 3 OutDrive vector strategies shown here. From left to right, there's a pan-T cell activation strategy, perhaps the most similar to the clinical benchmarks, but importantly, leading to much lower cytokine production with commensurate transduction efficiency. In the middle, a conditional activating approach, which can target CD8s or CD4s with dramatically reduced cytokine production relative to pan-T cell. And then all the way on the right, the non-activating approach, which, as we'll show you in a moment, is leading to minimal cytokine production and maintenance of a really exciting stem-like and naive T cell phenotype. This is the data to support what I just told you. On the left, you can see cytokine release. The benchmarks are up there. Those are the clinical in vivo benchmarks that I'm sure you're all aware of. What you can see for pan-T cell, conditional activation, and non-activating is about an order of magnitude reduction in cytokine release down to what's barely detectable for our non-activating approach. In the middle, we're maintaining high transduction efficiency that's equal or better than the clinical benchmarks, and you can see really, really great specificity in this case, targeting CD8s for conditional activating and non-activating. And then on the right, of course, the maintenance of that naive or memory phenotype. In summary, this is our pipeline. Lead program OPB101, IND is cleared, 3 clinical sites are activated, and we're terrifically excited about that program. OPB201, the follow-on program, IND submission on target for around year-end. And importantly, we've gone at Outpace from concept to development candidate, and from development candidate to IND for each of these programs in about a year for each of those cycles. So we move with exceptional speed. We're terrifically excited about expanding this pipeline over time. We were thrilled to close an oversubscribed $144 million Series B led by RA Capital and a wonderful world-class syndicate of investors last year. That will drive us through a durability readout for the phase 1 trial of OPB101, initial clinical and translational data for OPB201, and a development candidate for OPB301. Importantly, along the way, we'll validate 6 of those technology assets that we talked about in detail today in the clinic and have a number of others ready in the wings for additional translation. It's an exciting year for us, and thank you all for your time.